Assalamualaikum and hello. My name is Muhamad Farid bin Muhamad Zain. I am going to present about my FYP proposal, which entitled Effects of Black Soldier Larvae, BSFL, and earthworm feeding to the growth performance of betta fish, which is betta splendens. Firstly, we are going to look at the research background. The research is about comparing the growth performance for the different feeding trial of earthworm and black soldier larvae (BSFL) with fish meal as a control. Husbandary illusions, also known as the black soldier fly, is an earthworm that is commonly used as a supplement for fish meal. BSFL or BSFL juvenile carp that is enjoyed by both fish and chicken. High protein is one form of premium protein that will be extracted from the BSFL. High protein is said to have a high crude protein amount of content and an amino acid profile that is well balanced. BSFL is a natural, long-lasting and environmental friendly product. It is said to contain around 40 to 50% crude protein, making it a high protein source for animal feed. Next is the earthworm, or commonly known as Pyrenix excavates. There are different species of earthworm that are said to contain about 50 to 60% crude protein in dry matter and have a low ash content, which is desirable characteristic for a fish with ingredients. Earthworms or Pyrenix excavates have been found to be a good source of protein. According to a study, it is also a high ingredient that contains a high protein that used as a dietary supplement for some fish species. This tropical earthworm species, high reproductive rate and biomass production make it suitable for worm meal production. Next, we proceed to the problem statement. The first one is we are going to find a way to replace the fish meal as a feed ingredient, as there was a limited supply of fish meal. The second is we are going to compare the effect of two different feeds, which are black soldier larvae BSFL and earthworm to the betta fish growth. There are two objectives for our research studies. The first one is we are going to investigate the growth performance for the feeding trial of black soldier larvae and earthworm. And the second one is we are going to study the factor that can affect the growth performance of betta fish. For the hypothesis, we are going to say first is null hypothesis. There is no difference in the mean body weight of betta when comparing with BSFL as feed and earthworm as feed, which we are going to use the formula mean body weight of BSFL as feed equal to mean body weight of earthworm as feed. And the second one is the HA. There is significant difference in the mean body weight of betta when comparing with BSFL as feed and earthworm as feed, which we are going to use the same formula. Mean body weight on BSFL as feed is equal to mean body weight of earthworm as feed. Next, we proceed to the literature review. First is the fish meal. Fish meal is known as an excellent source of highly digestible protein or essential vitamins and minerals, long-chain omega-3 fatty acids, EPA and DHA, according to IFOMA 2001. For centuries, it was used as parts of animal feeds, but in the last 50 years, fish meal processing has become global enterprise. Next, we proceed to the second literature review, which is BSFL, or known as Black soldier fly. Black soldier fly, or Hamartia illucens, is a type of fly that belongs to the Stereomidae family. The cycle of black soldier fly will run parallel to the flow of organic waste transformation for the development of BSFL larvae. The adult fly is black in color but has a wasp-like appearance and measure 15 to 20 mm in length. The larva will grow to be 27 mm long, 6 mm wide, and weigh up to 220 mg at the age of their larva stage. Next is earthworm. Earthworms are known for their efficiency in transforming plant and animal waste into biomass, allowing them to be used as a feed ingredient in animal production. From a ton of animal waste, 100 kg of worms can be created. Earthworm is a high-quality protein that is used in livestock feeds, but the practical use of earthworm meal is influenced by economic. Vermicomposting produces protein-rich earthworms as a byproduct. It is said that growing earthworm is easier than raising an insect, both economically and environmentally, since many earthworm species are adapted to a wider temperature range than insects. Next is for the methodology. The first one is sample preparation. We are going to use 24 ornamental fish, which is betta splendors, and we will divide this into 3 groups, 2 treatments and 1 control, and in each group has 8 fish each. For the second one is fish feeding trial. The treatment 1, we are going to use fish meal as a control, and the second one we are going to use the treatment 2, which is we will be putting black soldier fly larva as a feed, and the treatment 3 we are going to use earthworm as a meal. And the third methodology is observation. First one, we are going to weigh the weekly weight of the fish. Second one, the growth rate of fish across feeding trial will be observed. And the third one is we are going to observe the correlation between growth rate and feed. And for the last methodology is data analysis. We are going to analyze the data by using one-way ANOVA test, which is SPSS statistic 64 software. For the expected outcomes, the result is expected to show a significant difference on growth performance comparing earthworm to the black soldier fly larva. Next is our milestone in our research. Our milestones is based on our Gantt chart. Milestones in the experiment of the effect of different fish on reproduction development of betta fish. The number 1 is the completion of research proposal, which is on May 2021. And the second one is the completion of sample preparation, which we are expecting to be done by July 2021. And the third one is the completion of feeding trial is around 2 months, and which we are going to expect to be complete in 2021 August. And the fourth is completion of water quality of observation, which is we are going to see on August 2021. And the fifth is complete data checking and analysis, which is going to be completed by September 2021. And the last one is by December 2021 we are going to complete the thesis draft and correction. That's all from me. Thank you for your attention.